Okay, this is lecture 15, and in this lecture we'll be going through a couple other signaling pathways, and in particular those involving ion channels and receptor tyrosine kinases. And the chapters this information's brought up in is 12.6 is the ion channels that we'll be talking about. 12.3 are the receptor tyrosine kinases. In particular, we'll be looking at the insulin receptor as our specific example of that. And then we'll be talking a little bit about cell division in this lecture and the regulation of cell division by protein kinases, specifically CDKs. And I should mention that the specific examples we'll talk about of ion channels are the sodium ion channel and the acetylcholine ion channel, and both of those are gated. As you might remember, we've talked about an ion channel before when we talked about transport through membranes, and that was the potassium ion channel. And in this channel here shown, remember we had these helices that had a dipole in them that kind of led the ions in the right direction. And you also had this selectivity pore where only potassium ions can kind of fit in there and get the hydrogen bonding interactions with that. Okay, this is an example of a channel that's sort of always open, but today we're going to be talking about those that are gated. Okay, and the gates on them can respond to a number of different things, in particular changes in membrane potential. We call those voltage gated, and then they also can respond to specific ligands on the receptor sites of these, and those would be considered ligand-gated ion channels. Okay. And as I mentioned before, there's many important examples of these, especially in the nervous system. And the two examples we're going to see today are voltage-gated sodium channels and the ligand-gated nicotinic acetylcholine receptor. Okay. Membranes they tend to be electrically polarized, and what that means is that you'll have, for instance, more positive ions on one side, more negative ions on the other, and then you'll get a voltage difference between the inside and the outside. And typically membranes are going to be negatively charged inside the cell compared to outside the cell, and the membrane potential is something like negative 50 to negative 70 millivolts. Okay. This potential is largely due to what's known as a sodium potassium ATPase. And what this does is it pushes 3 sodium ions outside the cell and allows 2 potassium ions inside. So you're getting 3 charges outside, 3 positive charges outside the cell, 2 positive charges inside. What that's going to do over time is it's going to make the inside of the cell, that side of the membrane, more negatively charged. And that's where the negative membrane potential comes from. Okay, this flow of ionic species across membranes depends on concentration gradients and the overall electrical potential. All right, so here's an example of our electrogenic sodium potassium ATPase. So you have a channel part that's in the membrane, and then you have a couple other pieces that take ATP and convert it to ADP so that it's using energy. This is an example of active transport. Okay, and while it's doing that, it can move 3 sodium ions out and 2 potassium ions in. Okay. And again, this generates that membrane potential. Okay. And in using that potential, chloride ions can move down their concentration gradient. That's a bad example because it's actually moving, these chloride ions are actually moving against their concentration gradient, but because the membrane is polarized, it's more negative inside than outside, they can move from inside to outside against a concentration gradient because they're moving down the electrical potential. Okay. Calcium, this is going high concentration to low concentration. Okay, that's moving down a concentration gradient. Potassium's higher on the inside than the outside, so that can move because it's being, pumped in by this system. It's going to be higher concentration inside the cell, so it can have, there's other channels like the potassium channel that we talked about when we talked about membranes in transport. Potassium can move down a concentration gradient to the outside, and finally sodium can move down a concentration gradient from higher on the outside because it's being pumped out by this, and then there's another channel for it to move from its higher concentration to its lower concentration. Okay. How do you calculate a membrane potential? Well, that's detailed here. I believe you have to do this on a homework problem, or at least I used to assign a homework problem like this. Not going to be a huge part of the class, but if it shows up on the homework, it might show up on the exam. And really, what you're doing here is you're using a modified version of a free energy equation. Okay, the free energy is going to depend, one, on the concentration differences between the inside and the outside of the cell, but then also on this factor. And this Z is the charge, F is Faraday's constant, and then you have a membrane potential. So this is sort of the electrical component, and this is the concentration component. Okay, at equilibrium, delta G equals 0, so we can set delta G equal to 0, and then we can solve for the membrane potential here, and then the equation can be rewritten in this way. Okay, and Z is the charge of the ion you're looking at. This funny script F is a constant, R is a constant, T is the temperature, and then you just need to know the concentrations inside and outside the cell, and you can calculate a membrane potential. Or if you're given a membrane potential, you could figure out, one of the concentrations. Okay, so voltage-gated and ligand-gated ion channels, these are pretty important in nerve signaling. So signals in nerves propagate as electrical impulses. So you can kind of think of networks of nerve cells as sort of like wires in a way, because they're propagating an electrical signal through them. Okay, and this electrical signal that goes through these nerve cells involves opening of voltage-gated channels, and particularly the sodium channel. Okay, that's kind of what we're going to say is the first step here. There actually is another step first where a signal has to originally come into the nerve cell, and then that will get the sodium channels to open. The sodium channels then propagate a depolarization of the membrane until they reach the end of the nerve cell where voltage-gated calcium channels can open. That allows calcium inside the cell. That calcium then triggers the release of a neurotransmitter. In this case, it's acetylcholine. Acetylcholine then opens a ligand-gated ion channel on the next receiving cell. Okay, so you have in this whole system, you have examples of two voltage-gated channels, the sodium and the calcium. And then you also have a ligand-gated channel, which is the acetylcholine receptor. Okay, let's look at a picture of this. Okay, and this starts here at, this is our step 1, where we have some sort of a polarization wave that moves down this nerve cell, and that started from some stimulus that this nerve cell picked up. So if you want to think of, we talked about how sight, that was with cis retinal going to transretinal, and then that creates a signal. So you can think of that, maybe as that was the stimulus, and then how that is propagated is you have this depolarization wave down the membrane that will eventually hit a voltage-gated sodium channel. Okay, that channel will then open. Sodium can come into the nerve cell, and you get depolarization. So there's a polarization wave. You can open, then this voltage-gated sodium channel opens, and you get depolarization. Okay, so that goes down, the nerve cell in a wave. Okay. Closely followed behind it, you get another voltage-gated ion channel that we don't really mention specifically, except here, and that's the potassium, voltage-gated potassium channel. And this acts, it follows behind, and it will actually open and allow potassium outside the cell, and that repolarizes the membrane. Okay. Right, so you have these two waves moving, very closely behind one another. Once this depolarization wave that you're getting from the opening of these sodium channels, once that comes to the end of this nerve cell, it will hit, the wave hits, and it activates these voltage-gated calcium channels. Okay, so once those open, calcium can come in. Okay. When calcium comes in the nerve cell, what that does is it triggers the release of these secretory vesicles. Okay, and they contain acetylcholine. They also contain some of the sodium and calcium that has been let in these channels. Okay, and those are released into the synaptic cleft. Okay, and they can travel to the next nerve cell. So that's what this is, the start of the next nerve cell. Acetylcholine will then bind to the acetylcholine receptor. Okay, so that would be step 5 here. It would be binding, and then acetylcholine receptor can then open, and that allows your sodium and calcium ions to go in, and that is what starts the next action potential, the next polarization wave. Okay. If we look a little bit closer, this is our voltage-gated sodium channel. Okay, and this is a very big protein. It has 4 domains. They're very similar to one another. Okay, each domain has a total of 6 transmembrane helices. So 1, 2, 3, 4, 5, 6. Okay, the important parts here are colored a little bit differently. There's a blue helix, transmembrane helix in each domain. That one is what's known as the voltage sensor. Okay, and that's helix number 4. Okay, between helix 5 and helix 6, there's this red loop, and that structure faces the outside of the cell. That red loop, these come together and form the selectivity filter. Okay, that allows only certain ions through. Okay, and that it makes up the pore region that the ions are going to move through of this channel. Okay, and then finally, helix 6, shown here in orange, that's what's known as the activation gate. Okay, that blocks the channel until a certain voltage is reached, and then it can open. Okay, it works very closely with the voltage sensor helix, and we'll see a picture of this that will make it hopefully make more sense. Okay, and then finally, this green piece here that's located between, on the inside of the cell, between domain 3 and domain 4, this is the inactivation gate that actually blocks the channel as well when it's supposed to be inactive. Okay, so this is how these, in the membrane, these 4 domains will kind of come together and make a sort of circular shape. And on the inside of that, the area between these domains is where the pore is, where the ions will move through. Okay. And again, the red portion, and we're not seeing these pieces in domain 1. We're kind of omitting them just so we can see inside. But these red selectivity pore, selectivity filter folds will kind of come together in the center there, and that is what filters out the different ions, only allows sodium to pass through. Okay, the voltage helices are actually can move up and down depending on the membrane potential. And by doing that, they allow the helix 6, helices number 6, to open and close and be the gate. Okay, and specifically activation gate. The inactivation gate has sort of a random structure, and it'll be open, and then in a certain amount of time it kind of closes again. You can think of it as sort of like a timer that this channel is allowed to stay open. Okay, so if we look at this ligand-gated channel when it's in the off position, okay, or closed, when the membrane's polarized. Okay, what we have when the membrane's polarized, it's more more negatively charged inside and more positively charged outside. Okay, that voltage sensor helix, they will be pulled more towards the inside of the cell because they have some positive charges in those helices. So they're going to be pulled closer to the inside of the cell. Okay, when they do that, the activation gate helices, helices 6, they are kind of smashed together and they close the pore. Right. When the membrane's depolarized, okay, and if it's depolarized, the charges are sort of equaled inside and out, or at least they're not as negatively charged inside. What happens is the positive charged voltage sensors will now move up. Okay, when they move up, it allows some room for the activation gate to kind of open up, right? Because we see it's drawn here in a cartoony kind of way, but you have this structure here that's a little bit more bulbous on the inside. Okay, and that's blocked by the voltage sensor when they're all the way down. But when those voltage sensors move up, that bulbous part can spread out a little bit more, and what that does is it opens that channel. Okay, when that channel's open, sodium can then pass from the outside of the cell to the inside. Right, this is what this looks like in real life. It's probably easier, right, to show these cartoon depictions, but this is just an example of what this looks like on the like a crystal structure of it. Okay, and you have this region here would be the selectivity filter, and if you look, it does make a very narrow hole there that only small ions can go through, and in this case it's very specific to sodium. Okay, so that's the voltage-gated sodium channel. Okay, an example of a ligand-gated channel would be the acetylcholine receptor. Okay. And specifically, it's called the nicotinic acetylcholine receptor. Okay. And this is an ion channel, much like the sodium ion channel. It lets ions go through, but this one, it turns out, isn't as specific. It can allows really all ions, but in specifically important are sodium and calcium ions to go through. Right. This is a ligand gated, as I said, and that's opened by the ligand acetylcholine. This has a little bit different structure. Okay, as we saw in the sodium channel, right? We have 4 domains, and by the way, going back even further, all 4 of these domains are connected. So this is one polypeptide. You have the N terminal here, C terminal over here. Right? But in the acetylcholine receptor, it's a little bit similar. You have these transmembrane helice regions, but that make up domains. But in this case, we have 5 domains, not 4. Okay, and we also, each domain is its own polypeptide. The N terminal going through to the C terminal. Okay, each domain has 4 transmembrane helices. Okay, they're denoted M1, M2, M3, and M4. M2 is the one highlighted blue or purple here. Those are sort of important ones to remember. And you have these outside regions that allow the acetylcholine binding sites to them. Okay. And a typical way to depict this in a cartoon version would be using these spheres. Okay, again, you have 5 of these separate polypeptide chains that are, you can really think of them as being identical, come together to form this complex, okay, 5-domain complex. And when it's closed, they have, and this purple is the M2 helix. Okay, so we're only showing the M2 helix here. The M2 helix kind of faces the inside of that pore. Okay, so this guy. Right, that M2 helix, when it's closed, the residues that point on the inside are sort of bulky hydrophobic residues. And that is what closes this pore or channel. Okay. The active binding acetylcholine, and specifically 2 acetylcholine molecules need to bind this ligand-gated receptor. When that happens, you get a rotation in these helices, these M2 helices, and the hydrophobic residues that were in the middle of the channel are now sort of buried, and you have a very small hydrophilic or polar residues that face the inside of the channel. So the channel is now open for ions to move, and they can interact with the polar residues that face in that channel. This is acetylcholine. Choline should be somewhat familiar because that was one of the head groups we talked about on phospholipids. And remember, choline was 2 carbons with this nitrogen trimethylated amine group here with a positive charge. Okay, acetylcholine just has an acetyl group attached to it. Okay, so that was our discussion on these gated ion channels. Okay, the next type of signaling system that we'll talk about are the tyrosine kinases. And these are what's known as enzyme-linked. Okay. They're actually fairly complex structures. they have a extracellular domain that binds a ligand and an intracellular domain that has some sort of a catalytic activity. And the specific case that we're going to talk about, the tyrosine kinases, that's their activity. And remember, kinase means an enzyme that phosphorylates something else. So what these do is they phosphorylate other proteins on tyrosine residues, and that tyrosine being phosphorylated is a little bit unique, as we've shown some other kinases that tend to phosphorylate on serine residues or threonine. These receptors are targeted towards tyrosine residues. Okay, the first step in this is that these add a phosphate group to itself. Okay, that's called phosphorylation. It's phosphorylating itself, which leads to a conformational change, which allows binding of target proteins to the catalytic site. Okay. And then once that happens, it adds a phosphate group to a tyrosine in a specific target protein. There's quite a few of these. They all have, slightly different structures. The one that we'll see an example of, the primary example that we'll use for this class is the insulin receptor. And that's this guy shown here. The INSR stands for insulin receptor. Okay. It has a sort of complex extracellular domain with alpha and beta parts to it. Okay. It doesn't necessarily matter that you know this structure. You just understand that this structure is responsible for binding insulin. Okay. The other thing I wanted to point out, that it's actually a dimer. You have, if this would be the alpha subunit and this is the beta subunit, you have two of those stuck together. So two alphas and two betas. Okay. On the inside of the cell, this blue region is the part that has the tyrosine kinase activity. Okay, so that is sort of repeated in these other tyrosine kinase receptors. There's GFR is the fibroblast growth factor receptor. A lot of these are the epidermal growth factor receptor, EGFR. A lot of those, that's what these are growth factor receptors. And the difference between these and the insulin, these are in a monomeric form. The insulin receptor is always in this dimer, but these exist in their inactive forms in monomers. When they bind their ligand, that's when they can actually go and dimerize with another copy of itself and then autophosphorylate. So it's easier to see this on the insulin receptor. When it binds insulin, that might activate this region. This will then phosphorylate this side. This side will phosphorylate that side, and then they both can phosphorylate other target proteins. And we'll see that again here when we look specifically at the insulin receptor pathway. Okay. Insulin is a hormone, and what it does is it tells a cell to uptake glucose and start the metabolism of glucose. Okay, this hormone, insulin, it's a peptide. It's actually made out of amino acids, a short chain of amino acids. It's produced in the pancreas. Okay. It's produced and released in response to nutrients, in particular glucose. So your blood sugar level, you eat something, those sugars in it, get absorbed into your bloodstream, your blood sugar goes up. that triggers your pancreas to release insulin. Okay. Insulin then travels through your system. It reaches target cells, in particular things like the liver muscle. Binding of that insulin to the insulin receptor initiates a cascade of events that eventually leads to increased glucose uptake and storage. Okay, so in normal people, when you eat something, your blood sugar will go up, fairly rapidly, and then also fairly rapidly you'll release insulin, and that insulin will tell your cells to take up some of this glucose, and then your blood sugar kind of drops back down to a normal level. It's important because glucose actually is somewhat toxic in higher levels, and it can cause a lot of bad things, and that's why people with diabetes, either diabetes type 1, where it's hard to make, they can't make enough insulin, or type 2, where they're desensitized to insulin. What that does is it creates higher blood sugar levels, and over time that can lead to some bad things happening. Okay, so one thing that this receptor controls is the import of glucose into the cell. So insulin will bind your insulin receptor. I guess I should start at 1. So in this state, I guess you could call this a fasted state. The glucose transporters, these molecules that allow transport of glucose into the cell, they are stored in vesicles. Okay, so they're not in the outside membrane, the plasma membrane. They're stored in vesicles. Okay, insulin will bind the insulin receptor. You get the autophosphorylation of each of these subunits. Okay, then they become active. They can phosphorylate other enzymes. That leads to a cascade, which eventually turns on the incorporation of these vesicles into the plasma membrane that contain the glucose transporter. Those transporters, when they're in the membrane, can then allow the glucose to travel inside the cell. Okay. Eventually, the insulin levels will drop. The glucose transporters are going to be removed from the plasma membrane by endocytosis. So these portions of membrane can kind of pinch off and be turned back into vesicles. And the transporters can then be stored for when they need to be used again. So that's one way in which this works. This pathway works to put a transporter in the membrane that allows glucose to be carried inside the cell. Okay, and once it's inside the cell, it's less toxic because it can be either used in the form of metabolism, glycolysis, or it can be stored in the form of glycogen. Okay. Another way in which these pathways work are by regulating gene expression. Okay, and that's through a signaling cascade. So insulin, this red dot here represents our insulin peptide. It binds to the extracellular domains of the receptors, and it activates the catalytic domain inside. Okay, so this extracellular domain is actually pretty big in this receptor. And again, it's a dimer, so there's 2 specific sites for this for insulin to bind. But here we show it only binding on one of the sites first. Okay, so once it binds, that switches on the catalytic activity. So the catalytic domain in one receptor, in this side, right, this side binds insulin, this is turned on. This guy then phosphorylates tyrosine residues in the adjacent receptor. Okay, so then it phosphorylates these guys. Okay, there's some structural changes shown here, and these are the tyrosines, the specific tyrosines that get phosphorylated. There are 3 of them. Not as important that you know their numbers, but knowing that there are 3 of them would probably be more important. Okay, that when they're phosphorylated, that causes a structural change that actually opens up the active site here, and when that during that structural change, what happens is it allows the target proteins to then bind with this, and then this domain, when it's active like that, phosphorylated, target proteins can bind, and then it can phosphorylate the target proteins. Okay, and the in particular, the target protein in this pathway that we'll talk about is a protein called IRS1. Right. And this insulin signaling cascade, right? The indirect interaction of phosphorylated IRS1. So IRS1 gets phosphorylated by the receptor tyrosine kinase. IRS1 then interacts indirectly with what's known as RAS protein, or RAS protein, RAS. RAS protein is an example of a G protein. Okay, so this is, it's not a G protein coupled receptor, but this G protein is used in this pathway. Okay, so IRS1, a phosphorylated IRS1, can then interact with RAS and actually cause a series of protein phosphorylations. Okay. ERK, or ERK, is one of the proteins that is phosphorylated in this cascade. ERK will then enter the nucleus, and it can cause the transcription factor ELK1 to become phosphorylated, and when ELK1 is phosphorylated, it becomes active, and it will stimulate the expression of specific genes needed in glucose metabolism or glucose transport, and one of those genes would be GLUT4, one of the glucose transporters. Okay, so showing this in a picture form would probably be easier to understand. Okay, so here we have insulin binding the insulin receptor. Okay, that will phosphorylate the other side of this receptor and make it active. Okay, then you can have insulin binding the other side, which would phosphorylate this side. Okay, once these are active and phosphorylated, IRS1 can bind, and when it binds the tyrosine kinase region of the receptor, it gets phosphorylated on specific tyrosine residues. Okay. When IRS1 is phosphorylated, it can then interact with these proteins GRB2 and SOS. Okay, when that happens, when it forms this complex, it then can interact with RAS. RAS then becomes active, and it can bind GTP. The activated RAS protein will then bind and activate RAF1. RAF1 phosphorylates a protein called MEK, MEK. MEK is phosphorylated on serine residues. Okay, so the tyrosine kinase is specifically in this receptor. Now we're getting into kinase cascade, where we're looking more like a classical kinase. So RAF is phosphorylating MEK on serine residues. Okay. MEK then can phosphorylate ERK, or ERK, and this is on threonine and tyrosine residues. Probably not super important that you remember exactly what residues are being phosphorylated on these guys. ERK, when it's phosphorylated, then it becomes active. It goes into the nucleus, and it activates and by phosphorylating ELK1. Okay, and ELK1 also needs another transcription factor, SRF, but probably not as important that you remember that. You can think of ELK1 as the transcription factor that it matters here. Transcription factor, what that means is it binds to regions of DNA that tell the cell, okay, express this gene. When that transcription factor binds, then that gene that it's binding on, or the gene after where it binds, will then be transcribed into messenger RNA and then translated into protein. Okay, so that's this sequence in a nutshell, and you can see that there's just thinking back to when we talked about the 5 characteristics of signaling pathways. Can we see some examples of those? And off the top of my head, this IRS1, when it forms this complex with GRB2 and SOS, that's sort of an example of modularity. Okay, it's forming a complex, sort of interchangeable parts, really, at these phosphorylated regions. Okay, it can also, for instance, in a different pathway, it can, form a complex, at another phosphorylated region. Okay, so this is an example of modularity. Okay, and then the amplification, right? So if we have one ROS protein being activated here, we're likely to get several RAF1 proteins being activated, and then each RAF1 protein that's activated can activate several MEK proteins, and you would get amplification at each of these steps, which gives you the kinase cascade. Okay. There are other tyrosine kinases. There are quite a few of these. The JAK-STAT signaling system, and these you will not need to know any specifics on. I'm just throwing them out there in case you're interested. People going into medicine might be, you might deal with these more depending on what field you get into. Okay, JAK is the protein kinase. STATs are signal transducers and activators of transcription. Okay, so you have, and one of these is the erythropoietin or EPO receptor. Okay, that EPO is a peptide hormone. It binds this tyrosine kinase, receptor tyrosine kinase, and it activates JAK, and JAK then phosphorylates STAT, and these STATs can then affect the expression of a gene. And so EPO specifically increases your production of red blood cells. Okay, and it's been used as a performance-enhancing drug, right? And the biggest example is in cycling. People like Lance Armstrong use this. He used it. One of the things he used to win 7 Tour de France's in a row, but he's not the only one. This is Bjarne Riis, and he actually, a couple years before Lance won his first tour, Bjarne Riis won a Tour de France, and the joke about him was he was called Mr. 60% because his hematocrit was 60% because he was taking so much EPO. Right. There can be crosstalk between tyrosine kinase receptors and specifically in G protein coupled receptors. Okay, so the example we use, the insulin receptor, Okay, that can phosphorylate IRS1. Okay, this IRS1 can then phosphorylate a protein known as PKB, which then phosphorylates serine residues on the tail of the beta adrenergic receptor. When those are phosphorylated, it signals that this receptor should be put into a storage, in a sense, in a vesicle, and taken out of the membrane. Okay, and if you think about that, does that make sense? Well, these are kind of competing pathways, right? Insulin being sent out tells you that you need to take up and store blood sugar. Beta adrenergic receptor is more that you need to, when this is active and it's being signaled by epinephrine, it's telling you that, okay, you need to liberate energy, you need to get glucose out of glycogen because you're going to need to use it. So if you have enough blood sugar already, you don't really need these beta adrenergic receptors to be releasing more glucose from glycogen. Okay, so they're kind of going in different directions in the metabolism and storage of glucose. Okay. Another example here is the insulin receptor can also phosphorylate a tyrosine residue on the beta adrenergic receptor, which then can couple other proteins, in particular, importantly, GRB2-SOS, which then activates RAS, which then activates RAF1, MEK, and ERK again, and then you get an alteration of gene expression. Okay, so it's not this activation of gene expressions isn't only from the activation of IRS1. You can also get that same endpoint by activating, in this case, our G protein coupled receptor. Okay. The final thing we'll talk about in this lecture are some cell cycle regulation. Okay, and these are done by what's known as cyclin-dependent protein kinases, or CDKs, cyclin-dependent kinase. And just as and refresh your memory, the cell cycle. Okay, you have certain phases of the cell, right? In these, you won't need to know these in the class, right? But the transition between these different cycles, you can look at the expression in the level of these specific cyclin-dependent kinases, right? They concentration in this case of this one goes up as you transition from G1 to S. Okay, and then it goes away, and you get another one that increases as you're transitioning from S to G2, and then again from G2 to M. Okay. And so how do these work? Well, they're regulated by phosphorylation. That's kind of been a theme of most of these signaling pathways. Okay, and also by proteolysis, and that's the breakdown of the protein by breaking specific peptide bonds. Okay, so if we look here at the starting point, our cyclin-dependent kinase in this state is inactive. Okay, there's no cyclin bound to it. It's inactive. Okay, the second step is where we have a production of a molecule known as cyclin, a small protein molecule. And synthesis of this leads to accumulation of it. It then can bind its cyclin-dependent kinase. Okay, this actually is still inactive because it's phosphorylated on a tyrosine residue, and specifically tyrosine 15. Okay, that blocks an ATP binding site. This is still inactive. Okay, it's phosphorylated again on a threonine at 160. Okay, that phosphorylation and the removal of the phosphate group on tyrosine 15 will activate the cyclin CDK protein. Okay, so once that's active, one of the things that it's a kinase, so it phosphorylates other things. One of the things it phosphorylates is a phosphatase protein. And a phosphatase is a protein that cuts off phosphate groups. So when it activates that, that can then go and cut off more of that phosphate group on tyrosine 15, and you'll see that that's kind of a sort of a cycle, a vicious cycle here, where you activate one molecule of your cyclin CDK, and then it can activate a phosphatase. That phosphatase then can go and activate many more copies of your cyclin CDK, and so you get a big amplification there. Okay. It can then go in when these active cyclin CDK complexes can then go phosphorylate other proteins that are involved in cell cycle regulation. Okay. Turning this off, that's shown here in the gray, how does that happen? Well, one of the things, once you get enough of these activated, they'll start phosphorylating this protein, DBRP. Okay, and what DBRP does when it's activated by adding a phosphate to it, it attaches molecule known as ubiquinin to the cyclin part of this complex. Okay, and that's shown here as ubiquinin. And it adds many of these to cyclin. And what that does, the ubiquinin targets that for breakdown. Okay, so things that have lots of that ubiquinin chain attached to them targets that, and specifically it's a protein that we're attaching ubiquinin to. It targets it to be degraded by the proteasome inside the cell. Okay, so you add these to it. That says, okay, bring this to the proteasome, and it's going to be digested. Okay, and when it does that, it removes the cyclin from your CDK, and then that turns it back off. Okay. So growth factors, and this is FYI, don't need to know the specifics here, but growth factors trigger transcriptional regulation of CDKs. And remember, growth factors, those tyrosine receptor tyrosine kinases that we saw, many of the ligands for those are growth factors. So growth factors will bind to their receptor tyrosine kinases. You then get a kinase cascade. The phosphorylation of proteins known as Jun and Phos will then activate your cyclins and your CDKs. You can also, that goes to activate other transcription factors and enzymes for DNA synthesis, and all those combined effects will give you, in this case, a passage from the G1 phase to the S phase. Okay, some other important cell cycle regulation activities. Okay, if you have a break in your double-stranded DNA, okay, that activates a protein known as P53. Okay, P53, when it's active, it's a transcriptional regulation that it will lead to the increased concentration of a protein known as P21. Okay, P21 acts as kind of a clamp or a cap on your CDK cyclin, and specifically this is CDK2 cyclin E complex. It caps that and it activates it, and then and what that does is it stops the cell from going into the next cell cycle and onto cell division. Okay, so it might seem a little counterintuitive, right? You're blocking a process of the cell, but that's important because if you have this big break in DNA and you go to try to replicate that cell, you're going to get an error that's because of that missing DNA passed on to the next generation of cell, which is not good. Okay, so this is really what P53 and P21 are doing, is to say, hey, we need to stop this cell from dividing and being replicated because its DNA is compromised, right? And that's important in people that have errors or deficient in P53 or even P21 are more susceptible to cancers because of that, because they don't have this mechanism, or this mechanism isn't working as well to stop a cell that has some damaged DNA from being replicated. Okay. And that's another way that the cell cycle and cell division can be kind of messed up is by actually damaged tyrosine receptor kinases. And so this is our epidermal growth factor receptor. And so this is the binding domain. Okay, it exists as a monomer. Once it binds that growth factor, then it can dimerize. And when it dimerizes, then you get these one side phosphorylates the other, and vice versa, activating those tyrosine kinase domains. If you have a case where you have this protein that's missing that binding domain, you can have a case where this tyrosine kinase is always on. Okay, it's never off. It's always on. It's constantly active in phosphorylating other proteins. Okay, and when that happens, you have no control. So it's just phosphorylating everything. Everything's on, and that's a situation where you again have uncontrolled cell growth, and that kind of is a clue that you're either cancer or going in that direction. Right? And that's why this protein kinase inhibitors are actually, many cancer drugs are targeting protein kinases and receptor tyrosine kinases to inhibit them for the treatment of cancer. Right, and this is, if we look at CDK2, this is our cyclin-dependent kinase. So this is not the tyrosine kinase receptor. It's our CDK, the cyclin-dependent kinase. Okay, this is what it looks like when ATP is bound, that binding site. And then this drug here, this anti-cancer drug, binds in that site as well, but it binds in a very much more tighter than the ATP itself, and in a sense it doesn't get let out of that active site, and it blocks it from ATP, which you need to become active. Okay, if ATP is not there, this kinase doesn't have a source of phosphate groups to phosphorylate the next protein in the series. Okay, so next time we'll finish up on chapter 12, and in particular we're going to be talking about integrins and some nuclear hormone receptors, and maybe mention a little bit about oncogenes. We sort of did that a little bit here today, kind of brought that up. Next lecture will be one of the shorter lectures in the semester, though. So I guess in terms of the exam, it won't have a huge effect on the exam as far as the amount of material, but I'm sure there will be, at least one question from the next lecture on the exam.